How is cancer cells evading the immune system? And the whole topic of immune evasion and how immunotherapy could be the next big thing in cancer research for patients. And I do get where they're coming from, because in aggressive tumours there is multiple populations of slightly different cancer cells. I totally get this. Distinct genetic mutations can accumulate in cells in different parts of the tumour, which then gives them unique subclones. For example, in glioblastoma, which is a highly aggressive brain tumour, there can be up to 6 different subclones, resulting in clonal heterogeneity. And then this is why therapies and drugs which may work on one subclone may not have an effect on another. But just as normal cells and new tissues sort of develop, it's not necessarily mutations that may be accumulating, but it's different differentiation of cells which are needed in order to provide the final picture, the final tissue which needs to be created. And you'll see what I'm getting at here in a minute. Tumours are dynamic, interconnected ecosystems, don't get me wrong. There's constant communications between cancer, immune and normal cells nearby, which would normally be, the stromal cells, the fibroblasts, macrophages, especially tumour-associated macrophages and cancer-associated fibroblasts, which are becoming increasingly important within the tumour microenvironment. But all this coming together, this crosstalk and this signalling interplay, this enables processes such as angiogenesis, the removal of their waste substances to occur as well. And when tumours interact with the immune system, they do have the ability to suppress its function. But this is only in transplantation assays and within transgenic mice within the lab. This is where this whole theory of immune evasion has stemmed from. But the fact remains that without the use of transgenic mice and the transplantation assays which are created within the lab at the benchside, this theory of immune evasion would never have came to light. I think it was Virtual back in the sort of scene that leukocytes were infiltrating the tumour right at the beginning, but this isn't necessarily saying that the immune system is going in to attack the cancer cell. It's associated with that sort of creating the cancer niche, that sort of inflammatory milieu, the inflammatory microenvironment, which is needed. The immune cells are needed to go in, cooperate with the stromal cells, and then form that cancer niche, which is needed inevitably to bring cancer stem cells from the bone marrow or another area of the body in order to create the barrier, create the cancer rogue organ. You will see that the immune system is also shown to aid in cancer initiation, progression and metastasis as well, and I'll link another paper to show that. But sometimes the immune cells may be directed to temporarily suspend or remove cell growth, don't get me wrong. Hence, at the time they are associated with good prognosis, this is maybe what they are doing, removing sort of dead cancer cells that are no longer needed. Moreover, in the lab, the immune cells, of course, they're going to directly kill and target transformed cells which have been transplanted into a host, into a mice or whatever it may be. But these transformed cells, they're not cancer cells, and of course they're going to ignite an immune reaction because they're seen as foreign to the new organism. Cancer stem cells, on the other hand, it could be just a recognition of self, which doesn't really need an immune response, especially if they're coming from the bone marrow, which is housing these cancer stem cells, housing these progenitor cells or embryonic stem cells, whatever they may be. It's not really clear from the research. But if the stimulus for cancer remains, there isn't a therapy in this world which is going to stop it. It's going to go about its business. It's going to go about creating the cancer niche. It's going to go about creating that barrier of cells which is needed for that particular organ. And destroying the bulk of the tumour, I can understand this. However, the bone marrow, which ultimately houses these stem or progenitor cells, if it's going to keep on releasing these cancer stem cells to the specific organ or to the specific area of the body, whether that be lymphoid or blood malignancies or solid organ malignancy, it's going to keep on performing that specific function, no matter if you destroy the cancer stem cells or the bulk of the tumour cells or not. So it really, evading host defences, evading the immune system, it's really only in transgenic mice models which they're able to knock out certain genes and sort of manipulate this response. The cancer stem cells, normal stem cells and embryonic stem cells and even normal tissues, they share over 70% similarity within their identification markers. So if we're going to direct immunotherapy against cancer stem cells, what are we really targeting? We're definitely going to wipe out a lot of our normal cells as well and make patients even sicker. I can't even think of the side effects, but it couldn't be great. Make sure to read the research notes, and I'll speak to you all soon again.